Welcome everyone to this optical education series, co-organized by the Optical Interesting Group at the Geneva Research Campus and the Australia Biomedical Group. Today's talk is the third lecture of this series, so it will be delivered by Dr. Jennifer Colonel, focusing on designing a sample-centered Bessel Beam selective plane illumination microscope. Jennifer received her bachelor degree in chemistry from Cornell and a PhD in physical chemistry from the University of Chicago. After a few years in the Silicon Laboratory at the Bell Labs, she joins a startup building a high-throughput line scanning confocal microscope for drug compound screening. She has been building instruments for biology ever since, including a commercial turf scope for single molecule DNA sequencing and array tomography system. She's currently working on extracellular electrophysiology probes in Tim Harris' lab at Geneva. So you will get a practical information on a specific bioimaging method, typically not covered in a traditional talk today, I believe. So this talk will be divided into two sections. So please mute yourself for the first presentation section. Then you can spit out your question in the second Q&A section. Jennifer, would you like to take over from here?
Sure. So I'm going to be talking today about a microscope that we built specifically to look at, to do high resolution imaging of whole mount Drosophila brains, so whole fruit fly brains. And it's a little bit unconventional, but I wanted to start off by mentioning sort of the forefathers of this microscope. This microscope is a direct descendant of the Bessel Beam SIM microscopes that were developed in the Betzig lab. And they're very nicely described in this Nature Protocols paper. And a lot of what I'm going to be talking about today, I actually learned from reading this paper. The structured illumination analysis that I'm going to be talking about, I got the code from Lin Shao. And he was actually here at Geneva at the time, and he helped me get started with it. And the best description of that algorithm is in his 2008 paper with Mats Gustafsson. So we're going to build a whole microscope, a whole imaging system around one sample. And so I wanted to spend a couple minutes to explain to you what that sample is. So each one of these guys is a whole Drosophila brain. This is the central brain and two optic lobes on either side. And what these images are showing you is the ability through very clever genetic tricks to get a single neuron, or actually two neurons, because one in each hemisphere of the brain, to express a protein. And you're only able to do this in Drosophila because you have this great genetic access. And of course, you can label the cells that way, but you can also put in ion channels to turn those cells on and off. And what that means is that you can activate and deactivate them and learn what how those cells affect the animal's behavior. And because we do circuits here at Geneva, the next thing you're going to want to know is who are the upstream and downstream synaptic partners of that cell that you learned how to activate and deactivate, so that you can reconstruct the circuit. So the whole point of this microscope was to build a microscope that could image these very selectively labeled tissue samples at very high resolution, so that we could determine if there are synaptic connections between those cells. So here's an example of sort of what that looks like in real life. So the ability to label specific cell types also allows us to label the synapses, the presynaptic sites in that cell type. This is a cell type that occurs in the fly eye, out in the lamina. It's called L2, which is not important. But because the fly has a compound eye, you get this nice repeating pattern of exactly one of these L2 cells per column. So the membrane is marked in red here, and the green spots are the presynaptic sites in L2 only. And so the essence of the measurement we want to do is we want to selectively label another potential postsynaptic partner to this cell in this tissue, like this fictional blue blob that I'm adding, and see if it co-localizes with the synapses that we've labeled. And then we want to be able to ask the question, does that co-localization of those labels imply that those are synaptically connected? And that's a question about what else could be in our voxel along with the two things that we labeled that might actually be the postsynaptic partners. So that's a straightforward question about the resolution of your imaging. and how much other stuff is in the tissue. So to see how much other stuff is in the tissue, you want to go look in electron microscopy where you've labeled everything. And I think that all fluorescence microscopists should look at an electron microscopy picture at least once a month to remind themselves that the tissue is full of stuff that you're not labeling. So in this case, this is in the lamina. It's one of the least crowded places in the fly fly brain. This L2 labeled thing is the red cell that we labeled before, and synapses are these little black blobs called T-bars. But what we need to focus on in this picture is what are the smallest things that might be in our voxel along with our labeled elements. And the smallest things are processes, which can go down to about 100 nanometers. And they can even get a little bit smaller, but we'll use 100 nanometers as our target for the smallest thing that might be in our voxel along with the things that we've labeled that might confuse our inference of a synaptic connection. So this is just a cartoon of that, showing that the worst case that you can have is if you've got a bundle of these processes going perpendicular to the largest face of your voxel, the direction that where you've got at least the lowest resolution. And for confocal resolution, along with the two things that you've labeled, your synapse and your potential postsynaptic partner, you're going to have something like 17 other 100 nanometer processes, or you don't know what's there. But if you do the calculations for the best possible Bessel Beam selective plane illumination microscope with 2-axis structured illumination, you calculate the smallest voxel that you might be able to get for a setup like that, given the NA that you think you can practically use, you get down from about 17 down to about 2. And that seems like a low enough number that for the most part, you're going to have, you won't have too many coincidental overlaps where you're going to incorrectly infer a synaptic connection. So that's what we set out to build, but I wanted to show you, or to remind you of what a wide range of options you have when you're looking at fixed tissue and you've got a high resolution imaging problem like this. And basically what I'm showing here is the Z resolution, which is sort of a proxy for resolution in general on the y-axis, and a very, very approximate notion of how long it's going to take to image and analyze your data on the x-axis. And in general, the more specific you make your labeling, the lower resolution you can image at and still infer something like a synaptic connection. And the less specific you make your labeling, the more analysis you're going to have to do. You get more information out, but it's going to take a long time to do the imaging analysis. So out here at the completely general labeling where you label everything is EM, and you'll be able to detect all of the synapses, but only after you do dense reconstructions of those complex EM images, and it takes a long time to image as well. And up at the other end with confocal and not doing any clearing at all, if you've managed to make a very specifically labeled sample, for example, if you've engineered your sample such that you only have synapses light up when you have two halves of a fluorescent protein bridging a synapse, so that only the synapses you're interested in are actually going to light up, you can image that at very low resolution and get the correct answer out. But it's a very tough sample to make. So you want to be somewhere in the middle where you're making a sample where the labeling is specific, but not so specific that it's too hard to make the sample, and the imaging and analysis is pretty easy. And so at the time that we were planning this microscope, xylene clearing plus a Bessel Beam SPIM-SIM scope struck a good balance. You did need quite specific labeling. You needed a specific label on those presynaptic sites. You needed a specific label on your potential postsynaptic partner. But then the imaging, once you built the scope, would be pretty easy, and the analysis would be easy. It wouldn't require detailed tracing or things like that. You can see from there's sort of a spoiler in this graph, which is in 2021, you have expansion microscopy, and that turns out to be an even better compromise with less specific labeling and fairly easy imaging as well. And I'll return to that at the end of the talk. So I wanted to talk in this talk about sort of some of the engineering lessons that we learned in building this scope. And that's what Chen was talking about when she said this was sort of an unconventional talk. So instead of just telling you about how amazing this microscope is, I wanted to tell you about some of the problems that we encountered as we built it. So hopefully you won't have those same problems when you need to build your custom system. So I want to talk a little bit about how we developed a custom immersion medium to match this very specific sample, a cleared Drosophila brain. I want to talk about we had to build custom objectives to work with that immersion medium and also to fit into the crowded space to make this 3-axis microscope. And so I wanted to talk about how that went and the mistakes that were made. Some of the tricks that were needed in order to accommodate those 3 objectives and the tiny sample space that you have left over. And then I wanted to also talk a bit about structured illumination analysis, because this this microscope is completely dependent on being able to do structured illumination, and then how that extends into working with Bessel beams. So starting off with clearing. So what clearing is it's removing the refractive index variation within your sample, for, within tissue in this case. And so the highest index stuff in tissue is usually membranes, unless you're dealing with stuff with bone in it. But for the most part, it's going to be membranes because they're fatty and they have a high index up around 1.55 to 1.56. And the lowest index material is going to be water. And so this was known long before I ever started working on it, but there's a standard clearing protocol for Drosophila tissue that consists of dehydrating the tissue by running it through an ethanol series to remove the water, and then replacing the ethanol with xylene. And xylene already has a pretty high index, about 1.5, but it does some magical chemistry that evens out the rest of the index variation in the tissue. And then you're left with a hunk of tissue, and you need to make a medium that will match it so that you can image through it. So this requires figuring out what kind of medium you want, but also quantifying what's happening. And so something that is a little bit tricky here is you wind up at an index that doesn't match any objective that you own. And so you have to ask the question, how am I going to quantify that? There's always going to be aberrations. How am I going to understand what's going on? So we built this kind of sample where we have the tissue mounted on a cover slip, and then we've added another surface to the sample where we can put beads as resolution targets, and then compare how those are imaged outside the tissue and through the tissue. And on the left here is a picture of what that looks like when you haven't cleared the tissue, because it's really easy to see the effect there. So without clearing the tissue, you can see that where we're imaging just through the medium, the beads are nice and clear, and they're round and they're bright. And when you're coming through the tissue, actually they're a little bit dimmer. That's hard to see in this picture. But at the edge of the tissue, they're super aberrated, they're stretched out, they're dim, they're a mess. And the reason why that's the case is you've got both scattering, and then the index of the tissue is way higher than the index of that glycerol PBS mixture. And so you've got terrible lensing at the edge of the tissue. This happens to be the edge of the optic lobe. If you go through this clearing process and you wind up with xylene cleared tissue, we figured out how to engineer a immersion medium, which is a mixture of dichloro and trichlorobenzene with an index of 1.552. And now the beads are imaged really nicely outside the tissue and through the tissue and even at the edge. So that's a really pretty picture. How do you actually get to that place? How do you tune the index of the immersion medium so that you get to that result? And it turns out that the simplest way to do it is to keep in mind that the tissue is acting like a little lens in there, so that it's got super high curvature because it's a small thing. I haven't really said this, but it's about 400, in its biggest dimension, it's about 400 microns, and then those little optic lobes are about 300 microns in diameter, so it's a very strong curvature. And what you want to look for is a focus offset imaging through your little lens, and not imaging through it, imaging in the medium here. And so what I've plotted in the bottom here, these are maps of the focus offset as a function of position in the image. And when the index is too low, this acts as a convergent lens. And when it's a little bit too high, when the medium is too high, then the tissue acts as a divergent lens, and then it gets more divergent as the index goes up. And this progression allows you to actually fit a point where you are switching over from being a convergent to a divergent lens, and you can optimize the immersion medium. So we wound up, as I said, at 1.552. And another little detail that I wanted to mention about picking your medium is that you really need to do this before you actually go and design the objective. So I didn't design the objective, but before you specify the objective for someone else to design, you have to know this index really well. And the reason why you need to know it really well is that any error in the index is going to give you spherical aberrations in your imaging. And for long working distances, this gets to be very severe very fast. And so what I'm plotting here is the RMS radius derived from ray tracing. So it's just a geometric radius as a function of error in the index for an objective designed for 1.552. And if you have a short working distance, like 400 microns, sort of more like a more normal objective, you can have a pretty big error, but those errors are still smaller than the area radius of that objective for 1.0 NA objective. And so you'll never see these aberrations. But if you need to build a 3 millimeter working distance objective, so you need to image through 3 millimeters of this tissue of this medium, your spherical aberrations get really big really fast. And so you can really only forward errors up to about 0.001. And so before you go to design your objective, you have to already know what that medium looks like. And there's one caveat there, which is if you have another way to correct your spherical aberrations, like you're going to build an objective with a correction collar, then you don't need to worry about this as much, but that's a much more complicated lens system and much more expensive. So we specified our objective, and with 1.0 NA and 3 millimeters of working distance and 1.552 for the index. And we got the first objectives back, and they were terrible. So this is what the point spread functions look like: terrible stretched out axial point spread functions, asymmetric lateral point spread functions. And this is recognizable as coma right away, which is usually due to decentering of one of the elements in the lens. And that's in fact what it turned out to be due to here. We found it really useful to actually characterize these things using a technique called phase retrieval to extract the wavefront error from the point spread function. So this doesn't come from an interferometer, it just comes from analysis of this point spread function. And that allowed us to learn that, yes, indeed, this is mostly coma. There aren't many other aberrations mixed in. And so we were able to communicate that back to our objective builders, and they were able to rebuild it, and it looks much better. It's symmetric. The axial point spread function is what we expect. And I wanted to show you what the wavefront error from phase retrieval looks like in this case, and it's flattened out quite a bit. But what I actually wanted to show you is it's much better, but you do still have these funny little high error points, mostly at high NA. And those are actually just a flaw in the phase retrieval technique. It's a fitting routine, and it has noise in it. And so you mostly have to look at what the general trends are across the wavefront error across the pupil that it has estimated for you. So how did that happen? I mean, it's great that we fixed it. It would have been even better if it had been correct the first time. And this was a rookie mistake, but you won't have to make it, because I'm going to tell you how to not make the same error that I did. So the rookie mistake was I didn't understand how these objectives were going to be tested and built. And so this is a pretty common way, I know now, for building an objective. You put it into an interferometer setup, and you're looking at light beam that the interferometer makes for you, going through your lens under test and coming back from a reference flat, and you look at interference fringes here. And actually there's an adjustable element in the objective that they set while it's in the interferometer to correct for the aberrations. And this works great, there's nothing wrong with that. But the problem is this is all in air, which has an index of 1. And so the effective NA of this setup, as they had it at the time, was only about 0.67. And of course, my imaging is it close to 1.0. That's where we had spec'd the objective. And the difference in NA there is just mostly due to the difference in index of the imaging medium. And so basically, they adjusted the lens so that it met the spec for a lens that you're going to run at 0.67 NA, but you need to be adhering to a much more stringent spec in order to have it operate properly at 1.0 NA. And so once they realized that, they were able to rebuild the lens for us, and it worked just fine. But the lesson for anybody out there who's going to build an objective is you need to understand this test and assembly procedure when you contract to build the objective, especially if you're working in an unusual medium like we are. So the wavefront error is pretty good. The other thing that you want to understand about your custom objectives is what the efficiency is of the transmission of high frequency information. So that's described by the optical transfer function, which is really just the Fourier transform of the point spread function. And what it's telling you is, compared to the DC band, how efficiently do I detect information out at sorry, this should be 1 over nanometers. My apologies. How will you detect information out here at 3/4 of the resolution of the microscope? The straight lines here are the ideal curve. And this is our actual lens measured on a couple of lenses with a couple of different bead sizes. And this difference tells you how different it is from ideal. And the important thing about this is you can compare it to a commercial microscope and see that a custom objective usually isn't quite as good. But the signal to noise ratio to get to a given resolution in your real images, where you don't have infinite signal to noise like you do for your resolution targets, is going to scale with the optical transfer function at that frequency. And the other thing you get to learn is where it bottoms out here tells us that the NA of these objectives is really around 0.9 instead of 1.0.
Jennifer, I just have a very brief question. So about the objective testing, which you just described, do you know by chance whether how can I say, whether at these bigger companies like Zeiss and so on and so forth, whether they use similar strategies, because they always announce their biggest and shiniest objectives with the highest resolution suitable for super resolution and so on and so forth. And I was actually, right now, I'm actually pretty shocked to see that, at least I guess with that specific company, that they use such a system where, how can I say, which is not at all suitable in order to test actually
I was going to try to brush over that, Ulrika, but
I'm sorry. Sorry about that.
So the answer is they had not built objectives at this NA before. I am certain from the performance of Zeiss and Nikon objectives, which I have now know how to test in some detail, that they are testing at the NA that they image at. And they have a big advantage in that they build whole imaging systems. And we had contracted this objective with an optics shop that doesn't build imaging systems. They just build compound lenses. And so they knew how to do the optical design. They're smart. They're perfectly good optics shop. But we had pushed them beyond their limit, and none of us realized just how far we had pushed them beyond their limit. And the reason why it was a difficulty is nothing is linear with NA. And so, so 1.0 NA is profoundly different from 0.7. Yep. And that's the other sort of lesson from that. Moving on, I wanted to take one slide to talk about phase retrieval because I found it such a useful technique. So what it is so usually you think about measuring wavefront error with a fancy interferometer setup, but in fact, it can be fit from your 3D point spread function. And I'm not going to be able to go into the algorithm for that here, but it's in this reference and it's a great paper. And what it is it turns out to be just a really great, easy general method for identifying and quantifying aberrations. And so if you've been building a lot of optical systems and doing things like fitting your beads to ellipses to try to decide how bad they are, this is a really interesting and useful replacement to try because it lets you get out all of the aberrations from a single fit. It's useful for doing things like comparing objectives. The other thing that I found that I really love it for is for centering things. So when you're trying to find the center of the field of view of your objective, you usually do that by balancing aberrations in different parts of the field. And so to measure the aberrations in different parts of the field, you can just image the whole field and fit those 3D point spread functions from across the field and see if the aberrations are balanced or not. So, so that's the end of the objective section, and moving on to sort of, all right, how are we going to get those three objectives and get a sample in the correct place question. So this is what the core of the microscope looks like. And so why are there three objectives? There's three objectives because of course you need a detection objective. And then we want to be able to do structured illumination. That's really key to our strategy here. We want to be able to do structured illumination from two directions. And we're going to be doing it by stepping the Bessel beam across the field of view. And in order to step the Bessel beam across the field of view in both X and Y, you need to be approaching from both X and Y so that you can step perpendicular to that direction. So we need these two excitation objectives and this detection objective all focused at the same place. And then we need to get the sample into the mutual focus of those three objectives. And then the other thing we need to worry about in the sample mounting is have we left optical access from all three of those directions. I'll show you a cartoon of that in a second. As far as making the whole thing usable, we worked with a really great mechanical designer, Jeff Jordan, who designed this really handy swing arm. So to make the sample mounting as compact as possible, we mounted the fly brains, that's these here, on tiny little cover slips and glued them to the end of 30mm rods so that we could cantilever them into that tiny space. And Jeff designed a swing arm that could bring that whole assembly into the space between the objectives really conveniently. It worked really well. So I wanted to show it off for you. And then, but the next question is, that space looks pretty small even physically, but actually most of that space is taken up with space that we need to leave empty. We can't have any non-matched index stuff in the cones that are in the cones that are formed by the incoming excitation beam and then the detection cone on the way out. So for example, if I mount the cover slip sort of perpendicular to the z-axis of these three cones, and the front edge here is going to wind up clipping the bottom of the detection cone, and that's going to cause aberrations so that you're not able to operate at the full NA of the detection objective. And looking from the back here, I know this might not be so easy to see, but looking from the back, we're clearing the excitation cones okay. But if we tilt that cover slip back too far, we'll run into those as well. So the basic message is we're going to need to control the angle of that cover slip. And this is actually pretty easy to do if you're willing to invest a little bit of mechanical design. So this is the flexure mount that holds the rod that holds the sample. We added a key to it and made a custom stainless steel rod with a flat that mates to the key. And then you can make a surface to mount your cover slip up here that has a controlled pitch and roll relative to the key angle back here. And so you can put that in with a very precise angle. And this is great because it lets you image all the way down to the glass. And that's particularly important because that lets you image beads on glass as your alignment sample. And frankly, you have to align the system every day. And so having an easy alignment sample is super important to making it practical. A really quick picture of the optical layout because we always like to show a picture of the optical layout. The things to note here are that we're making the Bessel beam in the simplest possible way. We just have a collimated beam that we're running through an annular mask. And then to switch between the two arms of the microscope, because of course we have to be able to send the excitation either to the right hand or the left hand objective, we used an electro-optic wave plate and a polarizing beam splitter, and that let us switch between the two arms without disrupting any alignment. And it also worked pretty fast too. It's switched in about 10 milliseconds, so that worked just great. Here's what the layout looks like in real life. The only message here is that it's not too big. It's only taking up about half of a large optical table, and it's really just not too complicated in general. So on to talking about Bessel beams. Why do we image with Bessel beams? And we image with Bessel beams because they have the super useful property that the length of the beam is decoupled somewhat from the size of the central core. So as we all know about Gaussian beams, you can focus them down to be quite small so that you have good Z resolution. But then the length of the beam is very short, and so your field of view is going to be very small. And you can make them bigger by not illuminating the whole pupil of your illumination objective, and it'll make it longer, but it'll also make it fatter. And so if you need to squeeze out really high resolution, that's not going to work for you. So Bessel beams, with Bessel beams, it's possible to make a long beam with a very small central core that's very uniform across the length of the beam. The beams that I'm using in this system are about 35 microns. So not huge, but longer than 10 microns. And but Bessel beams have a huge drawback, which is also very well known, which is only about half the light is in that central core that we really want to do the imaging with, and the rest of it is in these side lobes. And so those are going to create an optical sectioning problem, which I've illustrated here. So like all plane illumination microscopy, the lateral point spread function is being set by the NA of the detection objective, and the axial point spread function in Z is being set by the product of the excitation beam and the detection point spread function. So that's just what that looks like here. Here's the Bessel beam profile that we actually measured, multiplying by our axial point spread function. And you can see the stripes from the Bessel beam in there. So there's high resolution information in there, but otherwise it still has the shape of the original point spread function of the detection objective. And so beads are going to be all stretched out like this, and you could imagine deconvolving those. But as soon as you have things that are close together, which was the whole point of building a high resolution microscope, those are not going to be separable. And so the basic message is the side lobes are going to mess up. They're going to give you poor optical sectioning in Z. So this, of course, was a known problem. It also had a known solution at the time that we built the microscope, because in the Betzig lab, they had already sorted out that a really great way to fix the optional sectioning problem with Bessel beams is to combine it with structured illumination. And so that's what we're going to do too. So we're going to step the beam across the sample to create a pattern and use structured illumination analysis to reassign the light to the correct place. So this is just a qualitative picture of what that looks like. These little movies are swiping between Bessel beam with no structured illumination. So just sweeping the beam and Bessel beams with structured illumination, looking at beads and looking at a little arbor of synapses and looking at the membrane label of a complex arbor. And just qualitatively, of course, the axial resolution is much better, which is what we expect. The other thing that we sort of want to know about the combination of the microscope and the sample together is the clearing good enough so that we are able to image through significant depth of tissue and get the same resolution. And so we're measuring that by looking at a neuron that looks like this. And this is up at the surface of the tissue. This is down at 100 microns deep into the tissue. And I went through finding the smallest processes that we were imaging and measuring their diameter in the XZ and YZ planes. And what you're looking for in this picture is there any trend of those smallest processes that we're able to measure getting bigger as we go into the tissue? And there isn't much of a trend. They're staying constant between 200 and 250 for XY and about 300 for Z, which is what we expect for the NA and excitation and detection wavelengths. So this is all a success. The clearing is good enough that the Bessel beams are in good shape by the time that you get deep into the sample, and you're able to image as well. So structural illumination is so central to Bessel beam microscopy at high resolution being successful that I really wanted to talk a little bit about how it really works. So what is structured illumination? It's imaging with patterned illumination, which is going to transform high frequency information in the object into lower frequency information in the image. And because it's patterned, you only cover part of the pattern with any single image. And so you need to shift the pattern to cover all of the object in real space. And then you can reassemble that set of pictures into a high resolution image. And here's sort of the canonical structured illumination picture. You have a high resolution object. This is simulated data. I have a high resolution object, and when you illuminate it with wide field illumination and convolve with a point spread function, you get an image with no high resolution information in it for my simulated objective here. But if you take the same object and illuminate with sinusoidal illumination and convolve with the point spread function, you get this funny looking image. And what is this? What this is it's a picture of the beat frequency between the sinusoidal illumination and the structure in the object along this axis, along the x-axis. And so the two lessons to learn here is, so these are pretty similar frequencies because I set up the demo with both the object and the sinusoidal illumination close to the resolution of the objective. And so the difference between them, which is the beat frequency, is really low, and that's why there's just one stripe in this picture. And then of course, there's no high resolution information in the other direction. And that's why our scope has 3 objectives, is so that we can have patterned illumination along the other axis as well. But so this is a good existence proof that structured illumination can show the existence of high resolution information in your object, but you need to figure out how to get that information out. And so here's sort of a simple one-dimensional example to give you an intuitive sense of how that actually works for something that isn't just a periodic sample like sort of the canonical illustration I gave you there. So here's a simple one-dimensional object that consists of 2 pairs of point objects that are spaced just below the resolution of my simulated objective. So when you illuminate with wide field illumination, you're just going to get 2 lumps out, and you can't infer the underlying structure. But if you illuminate with sinusoidal illumination, of course, the image that you get depends on how that sine wave lines up to these point objects in here. And so the 3 traces in this picture are different positions of the sine wave relative to the field of view. Those are called phases in structured illumination. And so with the first one of those positions, you aren't getting much signal from either one of these objects. And that's because both of them are lined up with troughs in your sine wave. And when you shift the sine wave and take another picture, they're both lined up with some intermediate intensity in the illumination. And when you shift it again, you get most of the signal out because now you've lined up the peaks of the sine wave more or less with their 2 point objects. And so you can see that the information is there if you know the frequency of your sinusoidal illumination and you know the positions at which you took these images, you can see that the information is there to back out the existence and position of those 2 objects. So I wanted to show the math really quickly because it's actually very simple and for me helps flesh out what's going to happen when you do this analysis in real life. So your real space image is the product of your sample and your illumination and convolved with the point spread function. And its Fourier transform is the convolution of the sample with the illumination function and then multiplied by the Fourier transform of your point spread function, which of course is your optical transfer function. And the important thing here is our illumination function is a single frequency. And so its Fourier transform is only non-zero at 3 points, at 0 and then plus and minus the frequency of the sine wave. And that means that for each pixel in the Fourier transform of your measured image, it is a linear combination of the sample at that frequency at which you measured, and then these 2 shifted frequencies shifted by the frequency of the illumination. And when you take 3 pictures like that and multiplied by this phase factor here reflects where the sine wave is in the image. And when you take 3 images like that, you can build this set of linear equations where this is what we want to know. This is the information about the sample. This is what we measured. This is the Fourier transforms of the measured images. And we measured the OTF by taking pictures of beads. And so you can solve that for the information about the sample that you want to know. And the final outcome is that you wind up being able to solve for information about your image over a range that is the maximum frequency of your objective plus the frequency of your sine wave. And so you've extended the frequency range of your image out by the frequency of your sine wave. And sort of as a side note, if the illumination pattern is created with your imaging objective, which is sort of the classic setup for structured illumination, then the maximum K0 that you can create is equal to the maximum frequency of your objective. And that's where the limit of structured illumination being 2x the original resolution comes from. And then one more point to note about this is that in your reconstruction, you have to weight each frequency by the efficiency with which you detect it. And that means that your high frequencies, which you detect with lower efficiency, as you saw from looking at the optical transfer functions for these objectives, are going to be sort of overweighted. And so noise at high frequencies is going to be overweighted. And that has an impact on what your reconstructions actually look like. So this is simulated data again. And I just have the simulated signal to noise at 10 and 45. And this funny background ripple that you see is, in fact, due to exactly that. Any noise in the image at high frequency, because high frequencies are transmitted at low efficiency, their components get multiplied by a big number. And so you wind up with noise in the background that is proportional to that big number. And of course, if you've got better signal to noise, that background ripple becomes less important. Here's just an example of that in real life, looking at a low background and a high background image. And I've actually had to scale these so that you could see them both, but this has got twice as much background as this image. And I had to scale the corresponding reconstructions as well. And so there is some background ripple in X that you're able to see here because I have it scaled lower. But if you look closely at this image, you can see there's quite strong background ripple in Z. And that's due to, which you can't see in this image. And that's due to this extra background in the starting sample, which is getting translated into, that noise is getting translated into background ripple at our highest frequency. So for me, at least, this was always really difficult to sort of grasp intuitively. And so until I ran across this useful analogy. And the useful analogy is when you're taking color pictures with a black and white camera, which of course, as fluorescent microscopists, we do all the time, you take it with different filters and they have different efficiencies. And so in this little toy example, I made a 3-color image and I made up detection efficiencies where I made the blue really inefficient and made images using these imaginary filters with some baseline noise that would be like camera noise and Poisson noise. And the images that you would get on your black and white camera with those 3 filters are pretty bright in the red and green, and it's really dim in the blue because my efficiency is low. So if I'm going to do a reconstruction of the color image from these 3, I have to multiply each one of them by 1 over the efficiency of detection. And so I wind up with blue noise throughout the image. And what that is it's the noise in the blue image magnified by the big number I had to multiply in order to account for the low transmission of my imaginary filter there. And this is really an exact analogy for where that background ripple comes from in structured illumination analysis. So in order to summarize what the structured illumination is doing in this system, it's the thing to do is to calculate the optical transfer function, and that's going to show you how much were you able to extend the resolution of your image. And so you get the optical transfer function by getting to by taking the Fourier transform of the point spread function in the image. And so that's easy to calculate by taking the Fourier transform of our illumination, which is a sine wave. And so it's just these 3 peaks and convolving it with the Fourier transform of our point spread function. And you get an image that you get an optical transfer function that looks like this. And the frequency range has been extended by a factor of 2. And actually also have sort of more support at the edge of the original frequency range of the objective as well. And we're going to be doing that same calculation to understand what's happening with our structural illumination with Bessel beams. So what's going to change? That was a one-dimensional case with single frequency illumination, and nothing else is going to look like that. So what's going to change as we go to more complex cases? As you add dimensions, when you're adding dimensions either in Y just by illuminating, by rotating your sine wave pattern in the previous example, or in Z, you're just going to figure out what the optical transfer function is and add your shifted frequency information along that axis. And otherwise, the math looks really similar. A more complicated illumination pattern, like a step Bessel beam, is going to give you an illumination function that has multiple frequencies in it instead of just the single frequency in our little example there. And the Fourier transform is just going to be a sum over harmonics. But each one of those is going to be a sum, is going to be a delta function in its own right with a scaling factor. So here's what that looks like. This is another figure that I took from that Nature Protocols paper. And to do structural illumination with Bessel beams, we're going to take a Bessel beam and step it across the field of view using a galvo mirror. And when you, that's equivalent to convolving your Bessel beam with a periodic modulation function turning on and off. And you get this array of Bessel beams here. This is the Z-axis here, and this is the X-axis. You're sort of looking at the beams as they go across the image. And looking at that in frequency space, the Fourier transform of the Bessel beam is not just an annulus, because this is actually a Bessel Gauss beam, because it's got we're getting a finite amount of light through there. And so it's got a big DC component in the middle and then a bright annulus at the outer edge. And the Fourier transform of our modulation function is a series of delta functions at 1 over the spacing of the original modulation. And you get this picture out. So this is the Z direction, and this is the X direction. And if we follow, in order to get the illumination function in X and Y, we follow at Z equals 0 out toward from low frequency in the middle to high frequency out here. And you can see it's going to be a series of delta functions. It's dark and then it's just bright where at these harmonic frequencies. So it's going to be a series of delta functions that looks a lot like the one-dimensional example that I just showed you. Going in Z, it's more complex. You've got intensity at Z and then intensity out at the annulus. And it's a different looking function at each one of the harmonics. But we could measure it and then sum those up to get the optical transfer function. So this is what that looks like. So for XY, we're doing 11 phases of structured illumination here. And we do 11 phases because we need to space the beams out pretty far in order to keep the background low. And in XY, we have so I should back up a little bit. Now the optical transfer function is in 2 dimensions. It's still plotting, it's still showing the variation in efficiency of transfer as a function of distance from 0. So low frequencies are in the middle and high frequencies are out at the edges here. And so the original limit of detection for the objective is depicted with this red circle here. And the optical transfer function of the Bessel beam SPIM is depicted in white here. And you can see it's extended beyond the original diffraction limit of the objective, which is what we expect. In XZ, the change is even bigger. This is because, so this huge change in the resolution in Z is being driven by the width of the central core of that beam. So you go from very low resolution in Z to resolution that's essentially a little bit less than the diameter of the central core of that beam. So having built that microscope, and it's pretty much functional, and now I've tried to tell you about how the analysis works, we have to ask the question about whether the whole system works together. So we're able to label the synapses, but the question is, are we labeling them with enough label that we're able to detect them? Are we able to resolve them in place? And the place where we chose to test that was with, again, my favorite neuron, which is L2 but now looking at these rather dense arbors in the medulla. Again, it's a compound eye, and so each one of these is a repeated cell. It's a repeat of that cell type in the different columns of the fly eye. This is just showing a blow-up of one of those, where the little red dots are where we fit the objects to the size of the 3D point spread function for the microscope. And we're very lucky because we know how many of those synapses there ought to be. So there's a dense EM reconstruction that's been done for 7 columns in 1 animal for this arbor. And so we know that there should be about 200 synapses in there. And we're getting pretty close to that. And then sort of the exciting thing is that we're also able to, instead of just measure that in 7 arbors, we can measure it over 40 or 50 arbors and map how it looks across the optic lobe and show that there's fewer synapses for young flies. But basically the message is that this demonstrates good synapse counting for this combination of the BB-SPIM sim and the cell-specific synapse labeling system that we're using. And you don't know that until you measure it. And then we know from the voxel size that if these synapses co-localize with the membrane label of a second neuron, we can infer a connection with pretty good confidence. And so the question is, so that's awesome. And are we ready to do science? And the answer to that is, well, it's complicated because in the time that we put this microscope together, other things happened in the technology out in the world. And so this is the same plot that I showed you before. It's just here so that I can point to things while I talk. So 2 things that happened while we were developing and building this microscope and making it work is that there was a ramp up of a couple of really big EM projects, which gives you the data that you get out here, where we hired a ton of tracers and actually did a reconstruction of a TEM volume of most of the fly brain, and then also launched a big project to do FibSem over a pretty big part of the brain. And this data tells you about stereotyped synaptic partners in the fly brain. And so one of the questions that we were trying to address with this microscope was, if I have identified a neuron that I think is involved in a behavior and I want to know who his partners are, how do I find that out? And so with the completion of these fly connectomes in the EM, you don't have to make any sample to do that. You can go look it up in the connectome, and that's clearly the better way to do it. So that application was gone. And then the other thing that had happened is that expansion microscopy came along. And so it was sensible to ask the question if you could go with easier labeling. So these are hard samples to make. It's hard to do very specific synapse labeling, and then making your postsynaptic partner label just label one cell. Sorry, I garbled that a little bit. Making a sample where you have only a single postsynaptic partner labeled is also difficult. And so on this sliding scale, with a new technology in the mix, you might want to go for making less specific labels and imaging at higher resolution. But I'm just going to show you what problems we run into with the Bessel Beam SPIM scope if we try to do less specific labeling. And I'll run through this pretty quickly. But so the top row here is with our cell type specific labeling, no background. And so we're able to make pictures of the synapses that are very clear with very little background ripple to confuse counting. And if we try to label all of the synapses, which is on the bottom row here, we're still able to reconstruct it. And in fact, our resolution is sufficient to resolve them because there's only about between 1 and 2 per cubic micron. So it's well below our resolution limit. But the background means that we have significant background ripple. And we know that that's from trying to reconstruct on top of all of this background at low signal to noise. And this is enough to confuse the counting. So that with the Bessel Beam SPIM scope, the background associated with labeling all of the synapses is simply too high, and it's not supportable. The other issue is that it's easier to label a family of neurons than to just label one. But then if you want to know, for example, if this soma is connecting to something over here, you need to trace that soma's processes through this semi-dense tract in the middle here, and ask and then you know if a green labeled process out here belongs to this soma or to this soma, or actually to some that are over here. But if you look in detail, so this little movie is a movie of looking at the synapses, looking at the processes traveling through this tract here. And I don't expect you to follow this in real time, but I pulled out a few frames so that you can see that you've got cases like this where you have 4 processes, and after another micron, 2 of them, these 2 have merged into this blob here. And then after another micron, they have split like this. And we don't have any way to assign this process to either this guy or this guy because we had them unresolved at this point. And so if you want to do less specific labeling, you need to higher resolution to reduce close encounters. Or to flip that around, if you have a higher resolution measurement, you get to make the simpler sample where you don't have your postsynaptic partners labeled one at a time. So I'm not going to talk much about expansion microscopy, but it's such a different sample prep that I wanted to make sure that you realize that it's a completely different sample prep. This was developed by Tilburg and Chen, and this is a recent review article of theirs. And the samples that I'm going to be showing you sort of follow the top row here, where you label your, you have your sample fixed, and you've antibody labeled to the proteins of interest, and then you anchor those antibodies to a swellable polymer that you perfuse into the tissue. And then you get to swell the polymer by adding water. And this will expand the tissue, and it drags your labels apart. And you have to have also dissolved most of your proteins in the meantime so that you can pull them apart without the tissue tearing. And all of your labels pull apart, but they pull apart in an isotropic way. And this gives you a sample that is now significantly bigger than your original sample, and you can image it at lower resolution, but your effective resolution of the information that you have is higher. The actual samples that we're looking at are a little bit more complicated because they required multiple rounds of that to work properly. But this is data from Josh Lilbus, and working with Paul here at Genelia. And this is actually looking at the same place in the fly brain, because we had established that L2 was a really good a really good reference place to look and count synapses. So he's doing 8x expansion microscopy. He's labeling all of the synapses in the sample. And the big advantage of that is that with those synapses spread further apart, you can afford to have a lower resolution microscope. And that means that you can move from something like Bessel Beam SPIM to a lattice light sheet microscope, which has lower Z resolution, but the background rejection is excellent. And then it's also very fast imaging as well. And you have to ask the same questions about whether your label is surviving through all of that sample prep. And here's his data on that, counting up the synapses in that same arbor and comparing to the FibSem, and it's a good match. So that's not a guarantee, but he showed that it all worked. And then the other thing that extra background rejection allows you to do is to have high resolution imaging in dense samples, and that allows you to trace. And so his movie here is showing the cell that is purple in this picture, of course, was originally green, but it's far enough apart from all of the other processes in the sample that it can be traced. And in fact, Josh, working with some collaborators, has cooked up an automated tracing algorithm that can trace these neurons. But the fundamental physical thing that makes that possible is we've moved them far enough apart that we can image them using lattice light sheet microscopy, which has this great background rejection. And the resolution is, because the sample is now so big, everything is spaced far enough apart that you can do that unambiguously. So the measurement conclusions here are, if you need to know the standard synaptic partners in Drosophila, you should go look at the connectomes. And if you're doing something where you need to know about your particular sample, so Josh in particular is looking at different species of Drosophila, and there isn't any reference connectome for them, then the best bet today is to do expansion microscopy and lattice light sheet, especially because Josh and Paul have put in the effort to make expansion work really well in Drosophila tissue. So I'm not trying to sell you a microscope today, but I do want to try to sell you on some of the lessons that we learned in building this one. So one is that it's easy and doable and worthwhile to quantify how well your clearing is working, and this becomes even more important as your samples get larger. And when you're thinking about how well your objective works, you should think about both the wavefront error, which is what governs, the width of a point spread function that you measure with a bright bead and no background. And you also want to consider the optical transfer function, which is going to tell you more about what happens when you have finite signal to noise ratio. Structured illumination is a really great tool, and I feel like it's sort of underused. And if you're doing any kind of scanned beam selective plane illumination microscopy, you always have it there. So if you're working with a co-localization problem that just needs a little bit more resolution, structured illumination might work for you. And finally, just because I think it's so useful, phase retrieval is a really useful way to understand your individual optics and your optical systems. And finally, some acknowledgements and some references. I can't talk about everybody here. I mentioned Jeff. Dan is very important. He wrote the software that runs Bessel Beam scopes in the Betzig lab and actually lattice light sheet microscopes, and he did the modifications for my scope as well. I mentioned how important Lin was in getting me started with structured illumination analysis. Kai Wang introduced me to phase retrieval, and Wes gave a lot of super useful advice about how to build a microscope. And then one more shout out to Jennifer Jeter, who taught me how to mount fly brains. And I'd be happy to take any questions.
Thank you, Jennifer, for the wonderful talk. I was really amazed by the rich optical details about phase retrieval, structure illumination, and aberration with the long working distance objective. So which I didn't know in the past. So I want to yeah, let's move on to the Q&A section. I want to start with two of my questions. So you talk about structure illumination. So can structure illumination be combined with other microscope other than the Bessel Beam selective plane illumination?
It can be combined with any kind of selective plane illumination. It can be combined with, well, it takes a lot of work to build it into a wide field microscope, but of course that's where it was developed. There's a version of structured illumination working with confocal in the Airy scan that Zeiss builds. And so if you're just collecting your light in a confocal microscope with a photomultiplier, you don't really have anything that can work like structured illumination. But the Airy scan has actually a little multi-element detector so that they can detect, they can use the Airy pattern of the illumination as structured illumination. And the structure isn't very deep. It doesn't have very high contrast, but it does allow a form of structured illumination, and it pretty much works.
Cool. So you also mentioned single noise ratio impacts the performance of structured illumination. It sounds very critical. So in practice, so like for the user, so how can we decide? How can we decide the threshold? Okay, so that's the good single to noise ratio. We can use structured illumination or not?
That's a good question. And I wish I had done, of course, you gave that to me ahead of time. I've never done a modeling study to make a really clean threshold. So what structured illumination is going to do is it's always going to boost your resolution. So if you have a low signal to noise sample, that's just fundamentally low signal to noise, then you're not going to be detecting, you're not going to be achieving the resolution even all the way out to the limit of your detection objective. And so doing structured illumination will actually boost your ability to see resolution that's kind of within the nominal limit of your detection objective, even when it can't get all the way out to the ultimate resolution that your illumination could get you. And it would make an interesting modeling study, and I haven't done it. And so I can't give you a clean threshold. It will always boost your resolution some, because if you have a low signal to noise ratio sample, you aren't getting all of the resolution out of your original detection objective either. And so if you do structured illumination, you'll always get more resolution out. It might just not be out to the theoretical limit.
So I believe so many of our audience actually are not come from optics background. So if people want to use structured illumination method in their lab, so I mean, so how people can start, or
how can people start? Well, depending on what kind of microscopy you're doing. So there actually are commercial versions of structured illumination from, I think there's one both from Zeiss and from Nikon. And I am pretty ignorant about how they actually work, because like most people who build microscopes, I only run my own, or almost. So I'm sure you've encountered that. But there are commercial versions of it, and that's the place to start if you're doing wide field microscopy. If you're doing plain illumination microscopy, I feel certain that there are public domain versions of the analysis out there. And unfortunately, I don't know where they are, but I'm certain that they're out there. If any one of you really wants to do structured illumination, I will engage to do a search for you and tell you how to do it. But I know it's widely used enough that it must be out there.
Thank you very much. So any other questions?
Yeah, I have a question, and thank you for the talk. It's fantastic. I really enjoyed the index matching aspect in the first part of the talk. And I wonder, my question is for the expansion. So the expansion, I guess, broadens a lot of biological structures in terms of size. The tissue being broadened, doesn't that require some special objectives as well? And if that's the case, do you actually encounter index change? And going back, do we need to recalibrate the optics?
So for the ideal case, so it's a many-part question. So people who are doing expansion microscopy, for example, we built two lattice light sheet microscopes here at Genelia specifically to do expansion microscopy, and we're using the 2 millimeter working distance water dipping objective from Nikon, and that's big enough to do most of a fly brain, even after it's expanded to 8x, and it's big enough to do. So you can't do expansion on infinitely thick slices of tissue. It doesn't quite translate. And so for most practical tissue thicknesses that you can address, you'll be able to get there with that 2 millimeter objective. When everything is working perfectly, the gel is almost entirely water. And so the index will be pretty well matched. That said, we do have non-ideal cases where that's not true. So far, we haven't seen it so bad that it affects the imaging a lot. Actually, where we see it is that the incoming light sheet doesn't wind up where we expect. It gets bent coming into the block of gel, and it gets deflected up or down. As those samples get bigger, we are bound to encounter those problems. And sometimes you'll be able to fix them from the sample prep side, and sometimes you might need something to help you, adjust the index of your immersion medium a little bit, and then like that 2 millimeter working distance objective I mentioned has a correction collar, and so you can fix it from there. If you have built a sample where you can image some beads at the bottom of the gel, you can fix it from there.
I see. So I guess going back to expansion, do you mean that if someone was to try to use confocal or a multi-photon system for expansion samples, they would have to, I guess, look at using beats as a calibrator?
Yeah, I mean, usually most expansion samples are going to come out pretty close to the correct index. It is worth checking if you're trying to do this all at the, if you're really squeezing the resolution hard, then it's worth doing that. And in general, you're going to need a long working distance objective for that. But 2 millimeters is enough for most kinds of expansion samples, because I think if you make, well, I say that sort of glibly, but expansion goes out to 20x now. And you don't have to start with a very big sample for a 20x expansion to now be a very big sample. There'll be a big demand for long working distance objectives going forward.
So a last question would be on the, again, going back to expansion, is do you face issues on long-term dehydration? So, the samples have to be immersed. And
yes, they're immersed in water. Yeah, you image these with a water dipping objective.
And I guess, as far as I understand, hydrogels are not that stable over time, so they tend to shrink. And so the tissue would change as well. Am I right?
So the answer is it depends. So I'm not fully versed in everything that Paul and Josh did a lot of work to get that 8x expansion of Drosophila tissue to be stable over the imaging time. And what that tells you is two things. They got it to be stable, and it wasn't stable before that. And it was exactly the problem that you're describing, which is over time the hydrogel would shrink. And because these are big volumes, they can, they image over a couple of days, and that could be bad. I don't know all the details, to just be perfectly honest, but doing a serial expansion where you take your expanded gel and then cast it in another gel somehow helps to stabilize the gel so that you won't have that problem. But different kinds of tissue are going to require different kinds of optimization, and so it's nothing's easy, nothing's really for free, but but you can get there for lots of different kinds of samples.
Thank you.
Yeah, if anyone has questions, don't be shy and don't hesitate to raise up.
Yeah, you can unmute and ask questions. Not too much. I'll just lead on to the next question, which is, I guess, going back to Yurika's previous question on phase retrieval. I quite like what you have done, but from recollection, I mean, it's tough to calibrate the objectives. And what you're saying is that you can take the point spread function and retrieve the phase of that. I guess that's only specific to a given field of view. Am I correct? And if we were to look at a larger field of view, that particular function would change.
Yeah. So if you, but you get that information just by imaging beads in different parts of your field of view. So like that particular example that I showed you where I was plotting the astigmatism as a function of where I am in the field of view. I mean, I literally just have a big sample and it's beads in this corner versus beads in that, sorry, beads in this corner versus beads in that corner. Yeah, so that's all you need to do.
I see. And that takes into account just the system aberration, which takes into account the objective, but not the sample.
Your objective is going to make most of your aberrations. Not all the time. If you have, you know, the only other element that I can think of that, well, I've built a lot of optical systems, and you probably have too. So every element has screwed up at one time or another. But the objective is usually the main source. That and beam splitters.
Yeah, I share the interest with Steve and Eureka. I'm also, yeah. So the phase retrieval sounds very amazing. So from the perspective of users, think about, so people use microscope. So that is there any way to have phase retrieval standalone tool to calculate the aberration?
It's pretty simple code. I got the version that I used from Kai Wang, and I don't know if he ever put it up on GitHub. But it's, yeah, I mean, it's a pretty simple calculation. The paper that I cited explains the algorithm very explicitly. I don't know if they've got their code online. That's an old paper from 2011, I think. Oh, it's right there. Phase retrieval. Oh, 2004. Yes, they may not have put their code online, but there's like I just have a really simple MATLAB implementation. It's not a very complicated algorithm, and it's super useful. It is imperfect. It's a fitting procedure. The fit can fail, but compared to how I personally had been doing those kinds of things in the past, I think it's a real improvement.
Cool. So we will put your email, your contact on the slide.
We can.
People has.
If I get enough emails, maybe I will, you know, put it up on my GitHub.
Cool. So can phase retrieval apply to, generally speaking, all microscope, or is that a specific requirement?
It's really about wide field imaging, and because it's sort of assuming. I mean, what it's really doing is it's making a estimated pupil plane with wavefront error in it, and then generating the point spread function from that estimated pupil plane, and then comparing it to your measured point spread function. And so that's easy to do in wide field. It's a more complicated calculation that the current code and algorithm doesn't account for things like, you know, the illumination is actually confocable. And so like I test objectives in wide field, and I test optical systems. Like when I build lattice light sheet microscopes, I do the testing of the aberrations doing wide field illumination, so that I can see what the detection objective is doing independent of the excitation. I know you have a special interest in whether it can work for 2-photon, and I think it can if you are doing 2-photon with unscanned detection. So what we'd be mapping then is just the how tightly focused is the incoming 2-photon beam. But you would need to correct it for looking at the square of the amplitude instead of it being linear, of course. But I haven't tested any of that. So
That's cool. I may get more information. I may discuss more with you.
well, you know, we work in
about
the same
using the
building,
phase
so
retrieval for 2 photons.
it'd be fun to work it out with you.
Very nice. So if we
Okay.
don't have any other questions, yeah. So then thank you very much for today.
Well, thank you for the opportunity, and I'm disappointed to not get questions, but there it is.
I mean, you will get following up question.
Okay.
Once you
I
put
think
your
that's-
contact.
I think there's a lot that you had presented in terms of like the chemistry, the biology, and the optics. So I think people would take time to absorb and come back with you with questions. Yeah.
Okay.
Yeah, nice. Then see
Okay.
everyone in the next lecture.
Okay.
See
Thank you
everyone.
very
Thank
much, Chen.
you.
You stay
And
safe.
thank you.
Thank you.
Stay safe. Bye.
Bye.